These are the slides on the topic linkage. So, linkage is the tendency of genes located close together on the same chromosome to be inherited together during meiosis. Means linked genes are inherited together in the next generation. So, in simple words, like if two genes are close to each other on the same chromosome, they stick together during inheritance, like best friends who always travel together. So, how linkage works? Actually, every organism has chromosomes, and each chromosome has many genes which are arranged linearly. So, during meiosis, at the time of gamete formation, chromosomes can exchange parts, and this is called as a crossing over. But when two genes are very close, it's very hard for crossing over and to separate them. So, they are inherited together. So, when two genes are far apart, crossing over can separate them. So, they are inherited independently. So, that's the basis of linkage. So, linkage and crossing over are opposite to each other. So let's understand more about the linkage. So William Batson and R.C. Punnett, they noticed it while studying sweet pea. So they expected a Mendelian ratio 9 is to 3 is to 3 is to 1, but got something different. The traits were inherited together more often. So T.H. Morgan later on explained this using fruit flies in the Drosophila melanogaster and gave the concept of linkage as well as the crossing over. So there are many theories of linkage, but the most important is Sutton's hypothesis of linkage. So Sutton in 1902 to 1903 proposed that genes are carried on chromosomes and chromosomes occur in pairs. So genes located on the same chromosome tend to be inherited together. This was a first step towards linking Mendel's factors with physical chromosomes. So coupling and repulsion hypothesis, it was proposed by Batson and Punnett after their experiments on the sweet pea. They found two unexpected combinations of parental traits. So coupling phase means two dominant or two recessive alleles tend to stay together. And repulsion phase means a dominant allele of one gene and a recessive of another tend to stay together. So later Morgan explained this phenomena as linkage and crossing over. So you can see in this diagram that blue flowered and long pollen, which is designated by capital B and capital L, is crossed with the red flowered and round pollen, which is designated as small b and small l. So gametes of blue flower and long pollen will be capital B and capital L, and gametes of red flowered and round pollen will be small b and small l. So capital B, capital L is dominant, and small b and small l is recessive. So you can see that in F1 we are getting the capital B, small b, capital L, small l. So these are all blue flowered, long pollen, 100% dihybrid because of the dominant characters of the blue flowered and long pollen. So F1 results showed that blue flower is dominant over red flower B, small b, and long pollen, capital L, is dominant over round pollen, small l. So when we test cross, in test cross, what we do, blue flowered long pollen hybrid is crossed with the double recessive character red flowered round pollen. So genotype you will having the gametes capital B, capital L, capital B, small b, small capital L, and small b, small l. And in this one BBLL small, you are getting only one kind of gamete, that is a small l, small b. So in test cross progeny, what we found that out of 16, 7 are blue flowered and long pollen, which are as their parental one. Then one is blue flowered round pollen, which is due to the crossover, different from the parent one, new combinations. And one is red flowered and long pollen. This is again the combinations, new combinations. and the 7 will be red flowered and round pollen, which is of parental type. So in this way, the combination, we are getting the ratio 7 is to 1 is to 1 is to 7. It means we are getting the parental genotype 7 and 7, and the new combinations 1 and 1. So it is because new combinations are formed due to crossing over, and parental are remaining same in the progeny due to the linkage of the genes. So Morgan's hypothesis of linkage, it was proposed by Morgan, and he studied this in the Drosophila. The main key points are: genes are arranged linearly on the chromosomes. The strength of linkage depends on the distance between genes. It means close the genes, strong the linkage; distant the genes, weak the linkage. It means if genes are just located closer to each other, the crossing over is less and linkage is more. If the genes are located at far away or at a distance, then linkage is weak and more crossing over occur. So this is how you can understand the crossing over as well as the linkage. So mainly the linkage in this slide, mainly the linkage is described. So you can see Morgan's experiment on the Drosophila also. So the example is given here: gray or black body, or wing shape, normal or vestigial. So you can see that through the diagram here, the normal body and normal wings crossed with the black body and vestigial wings. So after combination in the F1, we are getting the normal body and normal wings. When it is test crossed with the black body and vestigial wings, we are getting the ratio as we are getting the parental types as well as the new recombinations. So new recombinations are found to be 8% or 9%, and parental type are found to be 46% and 37%. So normal body, normal wings means parental kind; black body, normal wings means black body, and normal wings means new combinations; normal body and vestigial wings means new combinations, and black body and vestigial wings are parental types. Parental type are 48% and 37%, and new combinations are 8% or 9%. So if you can see here, the more the genes closed, more the linkage occur. So this is how the linkage grow. You can study the linkage, and you can also study the chromosomal theory of linkage, which was proposed by Morgan. So genes mainly on the chromosome. Then you can see that linkage can be of two kinds: complete linkage and incomplete linkage. So genes which are very close, there is no possibility of crossing over. So they are showing the complete linkage because they are inherited together. So examples are certain genes in the male Drosophila and sweet pea. And in complete linkage, the genes are close but not very close, and crossing over occur, and crossing over can separate them. So examples are in female Drosophila, crossing over happens. So this is showing the incomplete linkage. So then you should know the linkage groups. So a linkage group is a set of genes that is located on the same chromosomes and thus inherited together. The number of linkage group is equal to the number of chromosome pairs. It means haploid number. Suppose Drosophila melanogaster having the 4 pairs of chromosomes, so it means linkage group will be 4. And human beings, suppose there are 23 pairs of chromosomes, so 23 pairs of chromosome means having the 23 linkage group. So each group represents one chromosome with all its linked genes. So this is the linkage. Then you can study about the factors which are affecting the linkage. Generally, you have seen in these slides that distance between the genes is very important. So lesser the distance, stronger the linkage. So crossing over frequency is very important. Higher the crossing over frequency, weaker the linkage. Sex of the organism is important. Crossing over is absent in male Drosophila, but showing the complete linkage. Then there are some environmental factors also, temperature, radiations, that can influence the crossing over. So you can see the factors of linkage and crossing over. Then what is the significance of linkage? Actually, it maintains the parental traits over generations, and it reduces the genetic variation in population, and it helps in constructing linkage map, and it is useful in studying chromosomal organizations and gene location, and it explains the deviation from Mendel's law of independent assortment. So you can just solve these questions. Who discovered linkage? And coupling and repulsion hypotheses were proposed by whom? And the linkage of number of linkage group is human is how many? And which type of linkage shows no crossing over? And strength of linkage is dash, that is proportional to the distance between the genes. You can solve these questions after just going through these presentation or going or understanding the concept of linkage. So this is how you can prepare your topic of linkage. So this is very good preparation, and it is very thoroughly studied and thoroughly taught to the students, because it's very important topic, and it is covering almost all the things about the linkage. So go through the linkage, understand the with the example of diagrams, and you can solve the questions. Thank you.